Hello everybody, welcome back to the Fundamental Pharmacy. My name is Sachin Jadhav, founder Fundamental Pharmacy, author of Fundamentals of GPAT Niper DI Pharmacist, Volume 1, GPAT Cracker Achiever MCQ book and few other books. Welcomes you in this YouTube video session. My dear students, if you are new on our channel, please subscribe the channel and press the bell icon so that you will never miss any video from my side. My dear students, in this session we are going to see very important part for your GPAT Niper 2023 Drug Inspector Government Pharmacist exam. Believe me, it is very important part and they have already asked a lot of questions from this part. So today we are going to see knowledge booster dose part 3, that is immunoglobins, that is a different kind of antibiotics and their important characteristics. So it's my request to you, please watch this video until end. My dear students, as you can see in this diagram, a graphical representation of your different types of immunoglobins or antibodies. So first of all, what do you mean by antibody? So antibody is nothing but the protein that is synthesized or that is released from our immune system against the infection. अगर आपको कोई infection हो गया, so infection is nothing but it is a foreign protein that will act as antigen, and against that your body or your immune system will synthesize antibodies, and these are nothing but the immunoglobins. They are nothing but the immunoglobins. So they are the globular protein known as the immunoglobins. So there are different kind of immunoglobins like immunoglobulin G, immunoglobulin E, immunoglobulin D, immunoglobulin M and immunoglobulin A. So these are known as a different kind of antibodies. So every antibody has a different role, every antibody has a different feature. So as you can see in this diagram, immunoglobulin G is a monomer, immunoglobulin E is a monomer, immunoglobulin D is also monomer, but immunoglobulin M is nothing but the pentamer. You can see 1, 2, 3, 4, 5. There are 5 units of antibody. They are joined together to form a immunoglobulin M. So it is a pentamer. Okay, and these immunoglobulins, they are joined together by this disulfide bond. Okay, so here also you can see this immunoglobulin A is a dimer. तो ये immunoglobulin G एक मोनोमर होता है, एक ही यूनिट होता है उसका. immunoglobulin G का भी मोनोमर होता है, immunoglobulin D का भी मोनोमर ही होता है. immunoglobulin M जो है 5 मोनोमर से बना है, इसलिए it is known as a pentamer, and the monomers are joined together by disulfide bond. Okay. As like other immunoglobulins, immunoglobulin A is nothing but the dimer. So ये दो मोनोमर से बना है, again okay, disulfide bond. So these are the diagram. We will see the individual features now. Okay, immunoglobulins or antibodies, quick revision or knowledge booster dose. So first, smallest immunoglobulin in the sense immunoglobulin having small molecular weight. So कम से कम molecular weight जिसका है वो कौन है? So immunoglobulin G has a smallest molecular weight. Then immunoglobulin M has a large immunoglobulins or antibodies, quick revision notes. So first is nothing but the smallest immunoglobulin, that is known as immunoglobulin G. Smallest means small molecular weight. Okay, so जिसका molecular weight बहुत ही कम है उसका कौन है? Immunoglobulin G. So smallest molecular weight immunoglobulin G. Largest molecular weight antibody, that is known as a immunoglobulin M. Immunoglobulin M is having largest antibody, largest molecular weight antibody. Maximum serum concentration having antibody. Which antibody will have maximum serum concentration? इसका सबसे ज़्यादा concentration रहेगा आपके blood में. So maximum serum concentration having antibody is nothing but the immunoglobulin G. So already they have asked the question on this largest molecular weight having antibody, that is the immunoglobulin M. Smallest immunoglobulin G. Maximum concentration किसका होता है blood में? Immunoglobulin G. Now minimum serum concentration, that is immunoglobulin E. So antibody having less minimum serum concentration or minimum plasma concentration, that is immunoglobulin E. Now heat labile antibody or the immunoglobulin which is destroyed by heat, so that is known as a heat labile. So immunoglobulin E is a heat labile antibody. Earliest to be synthesized immunoglobulin, that means the first immunoglobulin which is synthesized in your body upon immune response. So earliest to be synthesized immunoglobulin, which is responsible for the primary immune response, is nothing but the immunoglobulin M. Immunoglobulin M. Now secondary immune response is given by this immunoglobulin G. So immunoglobulin G is responsible for the secondary immune response. Which immunoglobulin will cross the placenta? So this immunoglobulin G that can cross the placenta. So it has the ability to cross the placenta and it will provide a passive immunity to the fetus present in the womb. Minimum half-life containing antibody. So immunoglobulin E will have a minimum half-life. Next is a maximum half-life having antibody. So immunoglobulin G has a maximum half-life. Remember, these points are very important for your GPAT, Niper, drug inspector and other exams. Then next is which immunoglobulin protects the surfaces of your defense cell. So that is nothing but the immunoglobulin A. So immunoglobulin A is known as a surface protectant. Okay. So immunoglobulin A. Warm antibody. So immunoglobulin G is also known as a warm antibody. Immunoglobulin G is also known as a warm antibody. We'll see the next point, that is a cold antibody. So immunoglobulin M is known as a cold antibody. It is also known as the largest antibody. It is known as a pentamer. It is responsible for the primary immune response. Remember, immunoglobulin which is present in the milk, that is immunoglobulin G and immunoglobulin A. So primary immunoglobulin A is regarded as a milk antibody. Okay, तो याद रखो. Now next is a immunoglobulin which fix the complement via classical pathway. So immunoglobulin M and immunoglobulin G fix the complements via classical pathway. Immunoglobulin A and immunoglobulin D, immunoglobulin A and immunoglobulin D, so that those fix complement via alternative pathway. So there are two pathway, classical pathway as well as alternative pathway for the complement fixation or antigen antibody reaction. Immunoglobulin G and A, immunoglobulin M and G, they fix the complement via classical pathway. Immunoglobulin A and D, they fix complement via alternative pathway. Immunoglobulin responsible for the primary allergic reaction. उसके ऊपर भी ऑलरेडी क्वेश्चन आ चुका है. So primary allergic response is given by immunoglobulin E. Or other sense you can say immunoglobulin E is responsible for the primary allergic response. Which immunoglobulin having maximum sedimentation coefficient? So immunoglobulin M is having maximum sedimentation coefficient. तो जिसका sedimentation coefficient ज़्यादा है उसका molecular weight ज़्यादा है. So that's why immunoglobulin M has a highest molecular weight and that's why it has a maximum sedimentation concentration. Remember. Next one is a immunoglobulin E will regain its activity. Immunoglobulin E will regain its activity. Now antigen recognition by beta cells is done by immunoglobulin D. So antigen recognition by beta cells. So for the antigen recognition, immunoglobulin D is responsible. Prouse-Neitz-Kustner reaction is given by immunoglobulin E. Prouse-Neitz-Kustner reaction is given by immunoglobulin E. Homocytotropism, that is given by immunoglobulin E. Milk antibody or immunoglobulin present in the milk, we have seen this already. Immunoglobulin A and immunoglobulin G. So particularly immunoglobulin A. So immunoglobulin G is present, can cross the placenta. इसके बारे में ये याद रखो. So generally immunoglobulin A is known as a milk antibody. Highest carbohydrate containing immunoglobulin. So immunoglobulin E will have a highest carbohydrate content. Lowest carbohydrate containing immunoglobulin. So immunoglobulin G will have a lowest carbohydrate content. So these are the specific important points. Now again we'll see in the details regarding all immunoglobulins. So types of immunoglobulin, then heavy chain. Okay, so these immunoglobulins, they are having two chains. One is known as a light chain and one is known as a heavy chain. So all immunoglobulins, remember every immunoglobulin will have same light chain. तो हर एक immunoglobulin में जो light chain होता है वो same ही होता है, but heavy chain जो होता है वो different होता है. Okay, so light chain gamma होता है उसमें और heavy chain हर एक में individual में difference होता है. So first we'll see immunoglobulin G. So immunoglobulin G, gamma heavy chain, G for gamma. Okay, now it has a molecular weight of 15 lakh dalton. It has a molecular weight of 15 lakh dalton. So it is having subtypes like immunoglobulin G1, G2, G3, G. They are the isotypes. Immunoglobulin G1, G2, G3. They are the isotypes of immunoglobulin G having molecular weight 1.5 lakh dalton. So immunoglobulin G molecular weight 1.5 lakh dalton. Now, what is the function of this immunoglobulin G? So it can pass through placenta and will provide a immunity to the child. Natural passive immunity. Natural passive immunity. It can transfer from blood vessel into tissue. It is very important for secondary response. Most abundant circulating antibody in the blood. ये सबसे ज़्यादा प्लाज्मा में होता है. You can see what is the T half of this 23 days. So it is having long T half. Now serum level of this antibody is 85% in the blood. Highest serum level containing antibody, that is immunoglobulin G, or most abundant circulating antibody. Next one is immunoglobulin A. So in case of immunoglobulin A, alpha chain is there. So G for gamma, A for alpha. So it is a monomer, and it is a monomer in the blood, but it is a dimer in the different tissue like tears, saliva, intestinal fluid. It has a molecular weight of 3,85,000 Dalton. Immunoglobulin A molecular weight 3,85,000 Dalton. Now, highest concentration of this antibody is found in the breast milk. So that's why it is known as a milk antibody. Immunoglobulin A milk antibody. Now it will also serve function to protect the bronchi and intestinal smooth muscles. So it transfers the immunity to the infants via milk. Now its T half is 6 days. इसका T half कितना है 6 days. Now it is present in the 7 to 15% in the plasma. Now let's come to the immunoglobulin M. So immunoglobulin M will have mu heavy chain. So light chain तो सब में common होता है. Heavy chain क्या होगा उसमें सिर्फ डिफरेंट होता है. G immunoglobulin G में गामा होता है, immunoglobulin A में अल्फा, M में म्यू. Okay. So immunoglobulin M will have mu heavy chain. As I have told you, it is a pentamer. It is a pentamer. 5 monomeric units joined together. What is the molecular weight? Molecular weight is nothing but the 9 lakh Dalton. 9 lakh Dalton. So highest molecular weight containing antibody. Monomers, they are held together by glycoprotein J chain, and monomers also have disulfide bonds. Remember, this antibody is produced as a result of primary immune response. सबसे पहले एंटीबॉडी कौन सा प्रोड्यूस होता है? इम्यूनोग्लोबुलिन एम. So produced as a result of primary immune response, responsible for the primary immune response and found in the high concentration in the blood. So highest concentration in blood में first antibody produced by fetus. इसके ऊपर भी क्वेश्चन आ चुके हैं. First antibody produced by fetus. Now its T half is 5 days. Now serum concentration इसका 5% रहेगा. ठीक है. Now immunoglobulin D means it is having delta heavy chain. What is the molecular weight? 1.8 lakh Dalton. Okay, molecular weight 1.8 lakh Dalton. It is found in the monomer, found on the surface of beta cell and acts as a receptor for antigen. So 2 to 8 days उसका T half रहेगा, 0.3% serum concentration. Now immunoglobulin E having epsilon heavy chain, and it is also present in the monomeric form. What is the molecular weight? 2 lakh Dalton. Now it is involved in the allergic reaction, and it releases the histamine, protects against the parasites. That's why immunoglobulin responsible for the primary allergic reaction, that is a immunoglobulin E. Okay, so these are the different types of immunoglobulin which is having very short half-life, that is a 1 to 5 days. So longest half-life having antibody, that is a immunoglobulin G, and shortest half-life having antibody immunoglobulin E. So it is present in the 0.02%. So highest concentration of antibody found in the serum, that is antibody G 85%. Lowest concentration रहेगा इसका immunoglobulin E का 0.02%. So these are the very important part. I hope you liked it. Thank you very much. My dear students, as you can see, you can also join our GPAT Niper 2023 online course. If you want such kind of full lectures, you can join our course where you will have live lectures, recorded lectures, study material, online test series. Everything is in 8,000 and you will have a 2,000 rupee discount. Everything in 6,000. We have very much promising result, as you can see in front of you, and you can order these books also from me. So every year consistently around 60 questions that can be covered in the GPAT exam, more than 100 questions that can be covered in the Niper exam from single books. Okay, so you can order these books also from me.